Hi guys, today's topic is probability in biostatistics. Firstly, we will discuss about its definition. Later, we will discuss more on it. So, what is probability? Probability is the numerical measure of the likelihood that a particular event will occur. It quantifies uncertainty and ranges from 0 to 1. 0 means impossible event, 1 means certain event. So, for an example of probability is likelihood of getting tail or head while tossing a coin. Formula of it is P equals to number of favorable outcomes upon number of total outcomes. Another formula that we will discuss more in when we will go deeper in the chapter is P plus Q equals to 1, where P equals to probability of occurrence and Q equals to probability of non-occurrence. Why it matters in biology? It enables researchers to predict disease outcomes and treatment responses, analyze genetic inheritance patterns, design and interpret clinical trials, make evidence-based medical decisions. Let us understand the probability based on an example. So, for example, there are 10 balls, 10 red balls in a bag, and 5 blue balls. So, what is the probability of getting red ball? We will get the probability based on our formula by applying the formula. So, it is number of favorable outcomes. That is, we want to see the probability of getting red balls. So, it will be 10 number of favorable outcomes upon for number of total outcomes. So it these two will be added, that is 15. So number of total outcomes will be 15. When we will cut it, it will be equal to 2 upon 3. So this will be the probability of getting red color balls out of the bag. Okay, P, in a bracket we will write R, that is red for red ball. So it will be our answer. I hope you understood the meaning and the example of probability. Now let's discuss some of the basic terms in probability. Firstly, it is experiment. So experiment is any procedure that produces observable outcomes. An example of it is testing a blood sample for a specific antibody. Next is outcome, a single result from an experiment. Example, the test result is positive or negative. So experiment is something we test something like that. It is called experiment, and outcome is basically a result. Next is sample space, the set of all possible outcomes. Example, positive or negative for antibody test, or something like if there's any test, then there's any another experiment, we get some positive responses, negative responses. It is a sample space, basically set of all possible outcomes. For coins, it is head or tail. Next is event, a specific outcome or collection of outcomes. Example, event A, test is positive, is one event from sample space. Next is let's discuss about types of probability. There are two types of probability, theoretical probability and experimental probability. Based on mathematical reasoning and known possible outcomes calculated without conducting actual experiments. Example, the probability of inheriting a recessive allele from heterozygous parents is theoretically 1 by 4 or 25% based on Mendelian genetics principles. So theoretically means it in itself it's saying that It is based on our mathematical reasoning or known possible outcomes. We do not conduct any experiments to prove it. Next is experimental probability. In itself, it's saying that experiments are being done. So its definition is based on actual observations and data collected from repeated trials or experiments. An example of it is in a clinical trial, 200 patients, with 200 patients, if 40 respond positively to a treatment experiment, positivity of success is 40 out of 100, that is 20%. So basically, when we will conduct any experiment or uh, something on 200 patients, and for about some medicine or treatment, if 40 people out of 200 respond positively or get well after getting the treatment, then the probability of the success of drug or medicine is 40 out of 200, that is 20%. Next, let's discuss about application of probability in biostatistics. So first is clinical trials. Probability helps determine sample sizes, calculate statistical power, assesses the likelihood of treatment efficacy versus placebo effects in randomized controlled trials, meaning that whether there's any likelihood of treatment, and determining the sample sizes for that. These things, probability in used in biostatistics. Next is disease risk prediction, in which doctors predict, or epidemiologists use probability models to estimate an individual's risk of developing specific diseases based on genetic markers, lifestyle factors, and environmental exposures. So basically, in disease risk predictions, uh, they they estimate or the risk of developing certain kind of diseases based on characters, based on lifestyle factors or environmental exposure, something like that. Next is genetic inheritance patterns. Mendelian genetics relies on probability to predict offspring phenotypes and genotypes, calculate carrier risks, consult families about hereditary conditions. So, in genetic inheritance patterns also, probability is used. Next, the foundation of biological analysis. Probability is indispensable, means very important in biostatistics, providing the mathematical framework for quantifying uncertainty in biological experiments, making informed clinical decisions under uncertainty, designing robust research studies, interpreting complex genetic and epidemiological data, advancing evidence-based medical practice, mastering probability concepts enables you to critically evaluate research findings and contribute meaningfully to the advancement of biological and medical sciences. I hope you understood the definition and its type and more about probability well through the lecture. In case you have any doubts or queries, then please feel free to ask it in comment box. Till then, bye bye and take care.